Okay, can you hear me? Okay, perfect. So thank you for having me here. So my name is Iker Núñez. I'm a postdoc at the Barcelona Supercomputing Center, which is the main infrastructure of supercomputing in Spain. And you might be wondering, so why is this guy talking to me about data scarcity in the era of data availability, right? So yeah, I will try to convince you that actually there are many scenarios in which data scarcity is a problem, and that is more close to most of the research we do than we think. So this is the outline. So first I will talk about the challenge of data scarcity and how we can actually overcome this in precision medicine, and how complementary data stored in large-scale databases can actually help in this process. Then I will go into some applications. Two of them are the interesting ones, let's say, because they were published. And the third one is currently undergoing, so I hope you also find it interesting. And then in the final part of the talk, I will go into the discussion and talk about how we actually addressed this problem and the main features perspectives that we have. So what is the challenge of data scarcity? So most of my work has been focusing on network medicine approaches, and particularly into bringing precision medicine into complex scenarios that are very well affected by data scarcity. Of course, precision medicine focused in trying to bring the P4 medicine paradigm, which is about having a more predictive, preventive, personalized, and participatory model. And this has been achieved in many cases by using computational biology methods like deep learning or network science that we are talking in this course. But this works really well in those scenarios in which we have a lot of patient data and many features from those patients. But when I don't have this, these models don't work that well. So basically, one possibility is to use complementary data, and we always talk about how important and how and the potential that we have in using complementary data and large-scale resources to try to fill in those parts of the puzzle. So the examples I will be talking about are mainly rare diseases, but you can think of other data scarcity problems in, for example, childhood oncology, or the stratification of cohorts by using some differential features, like, for example, sex and gender bias. Okay. So, of course, network models can help in analyzing and representing data of multiple types of detail. And basically, this can affect from very classic models like PPI or signal networks to more complex or more different data like drug interactions, or also enhance and promoter interactions. And what we need is a model for proper integration of these representations in network models. In our case, we have been working with multilayer networks, which are complex models in which we just have different networks representing some sort of data, and we have a connection between the different networks that we start to call layers. So in this example, we can have 3 different databases. One of them is protein-protein interaction, another one pathways, and the final one metabolism information. And all of them, I have representations of genes and their relationships depending on the database. Okay. And then I have connections. In this particular case, we connect a gene with itself across the different layers, and this is useful in the sense that allows us to analyze concurrent information across different layers. But depending on the problem that we would be interested in analyzing, your model would be quite versatile to try to represent the information that will be more important for you, of course. So then the edges that are connecting the different layers are interlayer edges, and those that are happening inside of the networks are the intralayer edges. For the analysis of these models, we focused on community detection because, as you will see in the examples, we try to find relationships between genetic information with causal information that is already known. So community detection is done in multilayer networks with, in our case, with an adaptation of the classical Leuven algorithm. But of course, it could be done with other with other algorithms, like, for example, InfoMap or anything that is done with random walk-based methods. In any case, all of them are limited by the problem of resolution, which means that actually with different levels of resolution in these community algorithms, we will find alternative community structures that are representing different important information depending on the scale that we are analyzing in the model. So with higher values of resolution, we will find more communities. With lower values of resolution, we will find less communities with more nodes inside. So what we observed is that actually there is a persistency of community membership in some of these nodes, and this is already known in network science to be manifesting some sort of a stronger topological relationship, because those nodes that independently of this disaggregating process are always together, of course, are more intimately connected. So our main question for this research is do the persistent communities have a more meaningful disease relationship, and is that meaningful in some disorders? So I will talk about 3 projects depending on the time. So the first one is on a rare disease, congenital myasthenic syndrome. Okay, so this is a disease of the neuromuscular junction. So basically, this is very similar to myasthenia gravis, but instead of being autoimmune, it's just that those same genes are mutated, and then I have a very similar phenotype to myasthenia gravis. So basically, those causal genes of the disease are very well known already, but this particular project is focused in trying to find genetic modifiers of severity, because the patients that have the same causal mutations are known to have huge differences in phenotype. So there are some with the same treatment do have a complete dependency during their life, and others that have normal life. So basically, this is the main goal. So of course, this is the structure of the neuromuscular junction, and the different genes are actually affecting all these different processes. Our target cohort, of course, is quite limited, and we have 20 patients with that share the same causal variant on the ChRNE gene, which is one of the subunits of the acetylcholine receptor. Okay, we have 8 patients that were labeled by proper clinicians as severe, and 12 of them not being non-severe. So how does our workflow work, basically? Our precision medicine approach starts by using DNA-seq data. So basically, we prioritize some mutations. In this particular case, we focus on compound heterozygous variants and copy number variation, because there was some initial segregation across the 2 groups. And then we want to find the relationship with the already known causal genes of the disease to know if there could be any one that of the mutated genes that are actually a modifier of this severity. Okay, so this is our input gene set, and we want to find if they fall on the same communities at different levels of resolution in a multilayer network model of the information that is more relevant for the disease. In this particular case, protein-protein interaction, pathways, and metabolism. Okay, so we compute the community structures at different levels of resolution, and we look for those modules of genes that are always together across this process. Okay, those modules, of course, you can imagine this is a disaggregating process. So we start with having a module of genes that are only connected in one value of resolution, then 2, and so on, until we just keep the module of genes that are always together. And in this particular module, what we do is to map the mutations of the different patients. And what we observe is that actually there is one module that is significant in terms of size. And in the severe patients, it's quite interesting because we observe a very classical scenario in rare diseases in which each one of the different patients has their own severity mutation, but all of them fall in the same process, let's say. And that particular process has a series of causal genes that are already known to be related to the disease. Of course, those mutations are completely independent of those that we observe in the non-severe patients. Then we go into the literature, of course, and we observe that the mutations that are falling in the severe patients are related to similar diseases and diseases that are actually overlapping with the phenotypes of the congenital myasthenic syndrome. And moreover, one of the genes was completely unknown to have any relationship with any kind of musculoskeletal disease. So we use this as a candidate for experimental validation. And actually, these experiments were done here in beautiful Canada by our partners of the Lockmuir Lab in Ottawa. And they actually observed that there was some reduction in the mutants of the US2A gene of the clusters of the acetylcholine receptor. Actually, the process that we observed to be related to this module was particularly that one that has to do with the presentation of the receptor, not the functioning of the receptor. So actually, this result is super striking. And also, there were some mouse experiments demonstrating the presence of this protein in the neuromuscular junction in mouse. So now we can formulate an hypothesis of how the severity works in the disease. Those patients, all of them had the same mutations that were falling in the subunit of the receptor. But the mutations of the severity are falling in the process that leads to the presentation of the receptor. So what we have is a defective receptor and then less receptor in the membrane. And this is causing the severity. Okay. This is super interesting because actually there are acetylcholine clustering enhancers, which are a type of treatment, are already known to have some effect in CMS severe cases, but there was no previous knowledge on why this happened. Okay. So this also talks about the potential in terms of giving explainability to drugs. Okay, that are observed in rare diseases but has no prior explanation. So I will go into a second project, and the third, I think we can leave it because of time. But this is more important, I'd say. So basically, this is one of those cases in which the follow-up comes 3 years before the original paper, which is the previous one. So that's science. So basically, in this particular case, we analyze a cohort of patients of medulloblastoma, which is the main childhood brain tumor. Okay. And we have a cohort of just 38 patients within the European project named Individualized Pediatric Cure, which is focused into analyzing childhood tumors. This is a very well described tumor in terms of molecular information, with 4 classical subtypes being already known. And this is the target cohort and the different multiomics data that we have that we can use as candidate prior to analyze with the multilayer network model. And basically, in this particular study, what we do is we try to do dimensionality reduction, because in this, when we do verification in this kind of analysis, we have huge dimensionality because the differential genes that are coming from each one of the different resources of the patients are many. So then doing any kind of downstream analysis like enrichment analysis or whatever, it's super difficult. So basically we have an original patient structure that was done with around 14,000 differential genes, each one coming from different multiomics data. And we come through another multilayer network, which in this case is not that specific of the disease, because we don't have exactly the same reference of why using these layers and not others. And then what we want to do is to get the minimal set of genes that actually is capable of recovering this to then use in downstream analysis. Okay. So here we define a novel concept in network science, which is multilayer community trajectories. And again, what we do is we compute community structures at different levels of resolution. And instead of focusing just in those that are always together, what we want to do is to optimize over this disaggregating process. Okay, that's why I say this is the follow-up. Basically, it's a very simple analysis in which each one of the communities are used as features, and then we compute a very simple amine distance to try to have a dendrogram of this disaggregative process. And then we define 2 different values with our theta and lambda, which correspond to the distance within the tree and the maximum number of genes that we find in the communities before discarding a given community. Okay. So in the end, what we do is a calibration of these 2 values. And we observe that it's for this particular cohort, these calibrated values were the best. And we're able to reduce the dimensionality up to 87% with a 94% of accuracy, Then we did a whole battery of robustness analysis. This is just a classical gene shuffling analysis. But then we also did an analysis of removal of genes and then recalibrating the different values. And we observed a first stage in which we are able to recover the information even with those optimized genes removed from the multilayer network. Then a second stage where we believe that basically we are finding the minimal information that we can find without those genes. And then a third stage in which we have eliminated anything that is meaningful, and basically we have no information related to the code, let's say. This is the patient stratification at the best pair with some p-values from bootstrapping. And what we observed is that there is we don't have 100% because there is a fifth group in our clustering that is already discussed in the disease biology, okay? We do a cohort validation in which we reapply this to another cohort with more subgroups, and particularly a subgroup that is happening between G3 and G4. And we observe that actually the accuracy gets even higher with an even higher reduction of the dimensionality. Again, as I said, the main idea here is, okay, now I have just 1,000 genes, I can do a network enrichment analysis. And as I have the networks, I can do this through the different layers of the multilayer and understand where does this information come from, right? This is the example for pathways, and we observe very expected results. I don't know, Sonic Hedgehog signaling is enriching Sonic Hedgehog, of course. But we do this for all the different layers, okay? And we observe drugs that are given to different patients and things like this. Then this third project, I will just jump it because of time. But basically in here is we do this, but we have not a prior knowledge of the subtyping of the patients. This is the next step. Let's do this completely unsupervised, right? So just to finish with the discussion, basically I gave you some examples of how we can deal with this data scarcity, but mainly we have the same approach for all of them. We have a modeling of prior knowledge coming from large-scale resources that we represent with the multilayer network. Then the mapping of the information from the patient into that model, and then the detection of communities at different levels of resolution and the analyze and optimization of this. Okay. So just to finish, I started talking about the objectives of precision medicine. So how does my work actually fit into that, right? Of course, in the first example, I was talking about the potential for the discovery of new targetable targets in very specific cohorts. And of course, the discovery and repurposing of new drugs, but also the explanation of drugs that are already observed to have an effect. And finally, this talks about the importance of data gathering and the analysis of overlapping diseases for rare disease research in particular. So I can jump the current research lines. So just to finish, I, of course, want to give a thank you to all the people that has been in these projects. Rare disease research needs collaborating even more than in other cases. And of course, to my group, the computational biology group led by Alfonso Valencia, and the machine learning for biomedical research unit in Barcelona. And of course, to all of you for being here listening to this. So I thank you, and any questions are more than welcome.
We maybe have time for one urgent question if somebody has one. Please just go to the microphone now. Yeah.
Thank you for your interesting presentation. So I have a question about the case for the patient stratification with the gene set. The parameter theta, and I don't know what is the parameter theta.
Okay, so theta is just the distance within the tree. So you have the dendrogram representing the disaggregation process, the process given by the resolution. So theta is just where you are at the tree. Let's say 0 is the end of the tree, so you will have all disaggregated, and then 1 is just one step, 2, 3. It's just like this.
Okay, thank
Okay.
you. Is it related to the final, how to say, the image you show in the final part of the presentation? You show the image of the with the networks based on the database, and patient will be indicated to the one.
But you
each
mean
cluster of
the patient clustering?
yeah.
Yes. So basically, for each set of values of theta and lambda, we have one clustering, and this is tested to be able to keep the original clustering that was used. Okay. So what I was showing was the best fitted values, but you can imagine that for each one of the different combinations of values, I'm testing if there is a proper recovery of the patient stratification. Okay.
Okay, I understand. Thank you.
Okay. Thank you.